And so our next session, I'm very excited to welcome, we're gonna move around the world today. Yeah, come up. Dr. Jones Gyamfi, who's gonna talk to us about my favorite subject, which is tumor microenvironments in triple negative breast cancer using single cell, of course. Thank you.
Thank you. Good afternoon. And my name is Jones Gyamfi from Ghana, and it's a privilege to present very early preliminary work on triple negative breast cancer. So breast cancer is still the leading cause of cancer in women, and while there's been significant progress in terms of treatment and cure, that treatment or outcome is not reflective globally. So in this map, if you look at Africa, still has a significantly high levels of mortality when it comes to breast cancer. And this is in part due to the fact that, yes, we now know that we molecularly stratify breast cancer, and unfortunately, the worst prognosis are in the HER2 positives and the triple negative breast cancers. And most of the tumors diagnosed in Africa are usually triple negative breast cancers. So there have been a lot of efforts in trying to subclassify triple negative breast cancers as a way of finding better ways to treat them. And a famous in layman in 2022 developed this way of classifying triple negative breast cancers further. One, by looking at certain key features that the tumors express. Whilst on average, globally, the incidence of triple negative breast cancer is 28%, in Africa, it goes up to about 40%, and in some places, like in West Africa, where I'm from, it can go up to about 60%. This makes it a very significant challenge in trying to understand why, because yes, it has a lot of, it's worse prognosis, there's no targeted therapy, and so the standard of treatment is still conventional chemotherapy. Recently, there have been a lot of interest in trying to use immunotherapy against triple negative breast cancers. One of those studies combined pembrolizumab with standard chemotherapy and saw significant improvement or extension in overall survival in women. Now, the effectiveness of immunotherapy really depends on the immune cells. So what types of immune cells are at play? Are they activated? Are they exhausted? Are they actively presenting antigens into the tumors and helping eradicate the tumors? And with genomic medicine, we know there's a significant underrepresentation from genomes from Africa. The TCGA still has about less than 2% of data from Africa. Much of the data there are from African Americans, which we now know doesn't fully represent the context of Africa. Yamachi Biotechnology, for about 2 years now, have made a lot of strive in trying to collect data on breast cancers in Africa and contribute data to the genomic databases across the globe. But still, we know that African American data is not reflective of African ancestries. Data from GWAS studies in kidneys have begun to show that, yes, there's similarities, but people in Africa still have very much huge diversity than African American. And so there's a need that we get data from indigenous African populations. So we decided to take a leap and jump the genomic data and begin to look at the tumor microenvironment in triple negative breast cancers from women from indigenous African communities by using single cell screening. And we basically wanted to understand what are the immune cells there, how is the tumor microenvironment looking like, what signaling pathways are activated in these tumor cells. So we collected, it's still actually ongoing. As of now, we have about 12 samples. So this data I'm presenting here just captures this small group of samples. Dr. Smith has been essential in helping us with this. If from the lectures we've had from Africa, collecting samples from Africa is really, really challenging because there's no standardized registries. And so for a huge part of this work has been Dr. Smith trying to organize this and have this done properly. And so as a way of further screening, it's when we get the samples, we test for BRCA mutations, and we look for those who have received treatment and those who have some form of, who have not received any treatment at all. Now, based on the numbers, yes, we look at the data comparing these groups, but it doesn't yield any significant differences because the numbers are still very small. So globally, there's quite a huge mix of cells in this tumor microenvironment in this population. comparing the normal to the tumors, we see that the epithelial cells population have a significant in the tumor compared to the normal. Immune cells doesn't show much difference in terms of numbers, but when you look at the details of it, you see some significant differences between the normal and the tumor populations. So looking at the epithelial population further, we basically put this into some major cell types to make the analysis initially easy. So looking at it in terms of major groups, we see that for the tumors, there's a wide array of different cells. So there's significant number of immune cells, and then there's a lot of epithelial cells. Whilst for the normal patients, there's a number of epithelial cells, but not so much immune cells. Stratifying based on whether there's BRCA mutations, whether there's chemotherapy, whether there's treatment naive, we see differences. But these differences, as I highlighted earlier, are not significant. We know that the numbers are small, so hopefully as the numbers grow. But looking at it in terms of the overall diversity of cells, we see that the tumor group seems to have a wide variety of different types of cells in the tumors compared to the normal cases or the adjacent normal sections. And so we basically try to compare and look at, is there anything going on in terms of epithelial cells? We see a very unique cluster of cells here in the tumors, which have a blend of markers related to lipid metabolism, related to calcium transport. It's still early, so we don't know what their role may be, but then we see this distinct classification. Going on the earlier chart I showed by Lehman, where they subclassify triple-negative breast cancers into certain subtypes. We decide to use those same markers to see if the triple-negative breast cancers can correspond to those patterns. And we see a very high level of the basal-like subtype 1, which has a significant expression of immune cell markers. So that is the blue group on the top there, accounting for almost about 68% of the cells that we see. Then we decided to look at specific epithelial cell markers. Key is that a subgroup of them express stem cell markers, so we see CD44 and CD24 expression. And then also there's some classical oncogene markers, MYC, EGFR, and there's HER2. There's been early reports that some triple-negative tumors express low levels of HER2, and so we see a few population of cells that still express HER2 in these groups. Then we decided to focus on the immune landscape, so classifying them just on the broad groups of immune cells, B cells and dendritic cells, macrophages. We see there's quite a significant differences in terms of the different groups, while the normal tumors have a huge chunk of T cells, the other tumor groups have a blend of them. Then we focus significantly on the T cells, and we find out they cluster into about 29 different clusters. But just focusing on some of the key immune cell markers, we see there's some population of cells that express this checkpoint inhibitors, CTL4 and PD-L1. And then we try to look at the general expression of T cell markers in terms of what they're doing. And so we find out that, yes, there's exhausted cells, and mostly the tumor cells have a lot of exhausted T cells, so T regulatory cells are high. Exhausted T cells that express certain checkpoint inhibitor markers are also high in this population. In terms of looking at signaling pathways, it wasn't surprising based on the high levels of immune markers in these tumor cells. We see a lot of pathways related to immune signaling. But of interest was that in tumor cells, there was a very low expression of signals related to activation of T cells. So yes, it probably corresponds to the earlier figure I showed, where there's high level of exhausted T cells in this immune cell population. And then there's high level of epithelial mesenchymal transition, MYC targets, as we saw when we look at the MYC targets earlier on in that picture. It's still very early on. We hope as we get data from our bulk sequencing, our genomic data, and combined with the single cell data, we get a better picture. We are also collecting data on exposures to certain pathogens. We now know certain pathogens also affect outcomes. So when we have all those pictures, we hope to compare our data with triple-negative data collected from other ethnic groups, other regional groups, in order to get a clearer picture of what is happening in our cohort or in our populations. Thanks to the incredible team, Novartis, for allowing us to divert some funds that was remaining to do this work, as we didn't have initial funding to start. Professor Kwabnado, who have led this initiative, and I'd like to mention Rosina Carr, who has been very instrumental in a lot of the analysis. Sadly, she has recently left us to do a PhD in the US, but we hope she'll still be able to contribute to the work we do. Thank you very much for your time.
We have a time for a question or two. So this is kind of my expertise, and we see this kind of in African-American populations. So given you see this T cell exhaustion signature, do you see any correlative kind of CAF phenotype or cancer-associated fibroblast differences?
We haven't looked at those yet. Yes, we see there's difference in fibroblast numbers, but as I said, we haven't delved into the details. We are hoping in the coming years we be able to recruit more patients and get the data to grow a little bit. This actually is about 100,000 cells in this analysis, so then hopefully we'll be able to delve into those type of analysis, look at the cancer fibroblast population and what they're doing.
And congratulations for starting the collection, because I know that's the hardest part. Yeah. But given you're paired with a wonderful pathologist, have you guys thought about pairing kind of the ERBB2 phenotype you're seeing in the single cell data to kind of canonical staining done in, are they staining for HERs and PRs and ERs, or is that not even happening yet?
That's not happening yet. There's no standard of doing this, right. So we have to initiate all of this. Patients would often have to decide if they even want their sample tested.
And we have to pay for it too, right?
Yes, and they have to pay for it too. So it's still early. Hopefully, if we get funding, then maybe we'll be able to do some of those things comparison.
Wonderful presentation. My question is, have you overlaid your single cell data with the normal breast data that are available in the HCA portal or the cell by Gini to see what difference do you see? Because you use the tumor adjacent normal, which is not typically normal. That already has a field effect. And the question she raised about the CAF, I just looked into your ZEB1 expression. ZEB1 is a clear marker of some of the CAFs in the normal breast itself, or not the CAFs, the fibroblast-like cells in the normal breast itself.
Are there any more questions? Well, you can come find him after the session. So up next, I'm excited to welcome, is the slide coming up? Dr. Ankita Chatterjee, who's going to be talking to us about smoking and how that's being affected in regions such as India. So, thank you.
Okay, a very good afternoon, everyone. My name is Dr. Ankita Chatterjee. I am from a research organization, JCMLRI, Kolkata, India. And today I will be presenting a part of our work on the AIDA consortium data on the effect of smoking habits in healthy population. So, smoking is one of the biggest public health threats worldwide. If we see the current prevalence of tobacco use from WHO, CDC, and World Bank data, we find that around 22.3% of the total world population practice smoking habits. It's much higher among the males compared to the females, with 36.7% in males and 7.8% among the females. Tobacco kills more than 8 million people each year, and the mortality rate, according to 2022 data, shows that deaths due to tobacco rank third after hypertension and air pollution. This makes the tobacco usage a significant public health. So there have been fragmented studies on the effects of tobacco intake on immune system, and most of them have been studied under a disease scenario. Some of the studies showed that nicotine, which is one of the hundreds of compounds that present in tobacco actually has a serious effect on immune system, and it causes immune suppression, and it's linked to neuroinflammation, multiple sclerosis, and atherosclerotic diseases. A recent study showed that tobacco intake causes heightened risk of inflammatory diseases. It has a long-lasting effects on epigenetic changes even after a person quits tobacco intake, and it causes change in cell states and function. Now, if we see, there are different organs that are affected due to tobacco intake, and tobacco intake is linked to multiple disease conditions in multiple organs. So there is a systemic interrelationship between them, and one way to study such systemic dysregulation is to study the effect of tobacco intake on blood immune cells. To do that, we are trying to study the question that, okay, whether smoking habits is affecting the immune system among healthy population, both among males and females. To do that, we are using the AIDA consortium dataset, which was generated from 5 different countries on 619 donors. We collected peripheral blood samples and we pooled them together to do single cell immune profiling using 10x 5' gene expression assay. The work has recently been published in Cell, and using the data, we tried to compare the differences in the cell proportions, cell types, and states among the smokers compared to the non-smokers. So the top figure showed the gross distribution of the different immune cell types, and the rightmost figure showed the distribution of the cell types in the same UMAP space among the smokers versus non-smokers. And we did not find any significant differences in the gross cell proportions between the two groups. In our data set, only 21.3% actually practice smoking. So this makes the data set for the smokers quite small. We looked into the cell types and states that are showing highest variance among the total population, and we find that plasmacytoid dendritic cells, cytotoxic CD4 positive T cells, granzyme K high gamma delta T cells, NK cells, these showed higher variance among the total population. So we checked the distribution of the cell proportions for these cell types across the 5 different countries, across 6 ethnic population, and we checked the differences after adjusting for the age, gender, BMI, and ethnicity. There was not much difference, but in certain cases there was a change in the proportion of the cell types among the smokers compared to non-smokers, and this showed variability across different ethnicities. Next, we look into specific cell states. So looking at the B cell states, we did a neighborhood enrichment analysis, and we found that naive B interferon high cell states showed higher enrichment of neighborhoods among the smokers compared to the non-smokers. We compared the proportion of this particular cell state across the population studied, and we find that the memory B interferon high population, the atypical B cells, and the naive B interferon high cell states, they showed significant differences across populations among the smokers compared to the non-smokers. And the interferon high cell state population actually showed higher proportion among the smokers compared to the non-smokers. So if we see the distribution here, The UMAP space doesn't quite visually distinctly showing the differences of the cell states between the smoker and non-smokers. But here we are finding certain differences over here and here, which are showing difference in the smokers and the non-smokers. Similarly, we looked into the CD8 positive cell states, and we find that here also CD16 positive NK cells and CD8 positive naive T cells with higher expression of interferons are showing increased neighborhoods among the smokers compared to the non-smokers. And this also came up to be significant when we compared the proportions across the study population after adjusting for age, gender, BMI, and ethnicities. So these are the cell populations here. This is the CD16 positive NK FCR1 GH high and LRC low IFN high population. This is the IFN high CD8 positive cell population. This is the CD8 positive, and this is the naive. This is the CD8 positive T IFN high population. And these are the differences between the smokers and the non-smokers. For the CD4 states, we again showed enrichment of the interferon high expressing CD4 positive cell population and the cytotoxic T cell populations. So looking at that, we then go back, and when we looked into the expression of the interferon signaling genes among the smokers and non-smokers, and we found that genes related, both the class 1 and class 2 regulated interferon genes are showing higher expression among the smokers than the non-smokers in the CD8 positive T cells population and the CD4 positive T cell population, but we didn't find this sharp distinction for the B cell population. For a subset of the data among the Indian population, we did a validation study using the Bioplex cytokine assay, where we compared the 39 different cytokines among 7 smokers and 23 non-smokers. The Bioplex assay uses antibody-based tagging of cytokine detection in the plasma sample. And we found that only three cytokines came up to be significantly different. Among them, IFN gamma is one of the cytokines that showed significantly higher expression among the smoker compared to the non-smoker, whereas IL-16 and IL-2 showed significant lower expression among the smokers compared to the non-smokers. So we know that interferon generally works in 2 main ways, in the type 1 way or the type 2 way, both working through the STAT pathway, but the downstream genes that regulate the interferon, that are regulated by the interferons, are quite different. Here, when we looked at the overall expression of the interferon-regulated genes, we find significant enrichment. But the question is, which part of the interferon signaling is mostly affected by the smoking habits? And that's the part now we are concentrating. Literature has shown that IFN gamma enrichment is found to be associated with smoking habits. And along with that, with reduced expression of the other cytokines, we can suggest that there is associated immune suppression effect of the smoking too. I would like to end my presentation by thanking all the study participants who volunteered to provide us the samples. I would like to thank Human Cell Atlas and Human Cell Atlas General Meeting organizers for providing me travel fellowship and allowing me to present my work. My institute, John C. Martin Center for Liver Research and Innovation, the whole AIDA team, and our team from India, Professor Arindam Moitra, Professor Partha Mujumdar, Supratim Ghosh, and Shumanta Sarkar. And finally, I would end my talk with the notion of say no to tobacco. Thank you so much.
Thank you very much. We have time for some questions.
Very interesting presentation. My question is, have you genotyped the smokers for CYP2A6? Because that is the main one, whether somebody is a fast metabolizer of nicotine or a slow metabolizer, that influences how the smoking affects their health. Do you know, particularly when you're comparing the different ancestry ethnic groups, maybe the CYP2A GWAS may completely affect the outcome of the smoking?
That's a very interesting suggestion. We did genotype all the individuals, but I haven't checked the difference in the distribution of the CYP2A gene genotypes across the across ethnicities among the smokers versus non-smokers. I will check it. Thank you for the suggestion.
I have a question in terms of, is it your goal to expand cohort? And if that's the case, is there ability within the metadata of the single cell to actually extrapolate how long you've been smoking and stratify within that? Because is it also an exposure thing that you're not able to see any, because you've collapsed across all the kind of different countries, whether or not you could include, as you include more samples, would you also include length of exposure?
Yes, again, a very interesting question. So we do not have the data of the duration of smoking for all the individuals. We have a very partial data, so essentially that's not enough sample size to compare it. But the literature reviews that says is that a person the longer the duration a person smokes, the way it affects the immune system could vary. Okay, so initially it could suppress the immune system, but it also leads to the healthy immune system prone towards autoimmune diseases. So that's an interesting hypothesis that could be tested later on. Thank you.
Thank you. Any more questions or on to our next speaker? Thank you very much.
Thank you for listening.
It's my pleasure to introduce a very good friend of mine. I know a lot of you in the room know him, Dr. Quan Nguyen, who is a king of all our applications and some of the tools I know many of you are using in the room to think about computation across populations. So welcome, Quan.
Thank you, Jasmine. Thanks a lot, the organizers, for the opportunity to present our work, and it's been a great conference as always. So in this talk, I will focus on 3 messages. The first one is how we can collaborate on generating a lot of spatial multimodal data to understand the biology better and to cross-validate the findings. The second one is about how can we make the spatial information useful for more people, essentially democratizing the spatial data, bringing it to more users. And finally, I want to introduce another consortia and call for the alliance between the collaboration between the different consortium on spatial omics. So before I start, I want to acknowledge the key members in my team who produce the data that I'm presenting next. So I'm using skin cancer as an example of the multimodal analysis, but this can be applied to any system. And as Jay mentioned yesterday, skin is a very interesting organ. You may know that it's the biggest organ, and it's about 3.5 to 5 kilogram organ, so surprisingly big. The spatial structure is quite defined, so there are 3 layers in the skin, and the skin cancer has 3 types of very common cancer, BCC, SCC, melanoma, and they have a very distinct biology. Problem is, what are the biology that make the cancer more deadly, more metastatic, or more common but less deadly? So we try to tackle that problem by looking really deep into the spatial multiomics of the skin. So we look at 24 patients, including healthy donors, and we apply 12 different technologies ranging from single cell sequencing to spatial imaging, spatial sequencing, and it covers different resolutions and different modalities as well as different number of metabolites or proteins or RNA we measure. And I will demonstrate that integrating these data together really help us to draw very high confidence biology. So in order to generate that kind of data, we need a lot of technology together. We're fortunate enough to have a good collection of tools in our lab, but there are always many other ways to collect the data through collaborations. So this is an example of two different types of data out of that 12 modalities, 12 technology I presented. We have a single cell data on this side and spatial data on the other side. Color again showing different cell types, and those came from two different samples, not like one tissue section and one UMAP and one and corresponding spatial map, but it's from two tissue sections. But we could see that the skin biology could be clearly seen through the layers of the hair follicle, and we are looking at the distribution of fibroblasts and the keratinocytes in the skin spatial context can be displayed in the UMAP context of the single cell, but when we combine them together, we'll have complementary data to validate the cell markers. And this is an example of how we combine three modalities. In this case, metabolite on the top there, in the middle is proteins, and then at the bottom is transcriptomics. So we can focus on the green cluster in the tissues section. So these are three different sections, three modalities, and we can combine the information to see what are the pathways that can be supported by three modalities. And in this case, we could discover that pyrimidine metabolism is a key upregulated pathway in the melanoma community. So by combining orthogonal data, we can come up with a very interesting discovery. Next question is, a lot of spatial features are known and novel. How can we get confidence that these features are reproducible? If we look at new patients or new cohorts, do we discover the same pattern again? So our team, we came up with a way to integrate the data across patients as well as across technologies. In this case, we try to tackle one question, that is to integrate the data to understand about cell-to-cell interaction. So we could normalize the data across samples in one technology, and we can also normalize the data across different technology. The results of that is that we can come up with a high confidence network of cell-to-cell interactions, and this network can be compared so that we can answer the question of what are the differences between the interacting cells in one condition compared to the other condition. I'm showing the integration of three technology here, Visium, Cosmix, and Xenium, and in each technology we have biological replicates. And when we combine the data together, we could come up with much cleaner network of cell-to-cell interaction. And once we made the comparison, now we can come up with robust patterns that can be observed across technology, and we can see at the y-axis on the far side there, and across cancer side, cancer sample on this side here. So we can see like one example of the IL-34 and CSF1R interaction between macrophage and melanocytes. We could zoom in and see the co-expression of the two, the ligand and receptor in the space, either by zooming in a small region or looking at it across a global map of the whole section. And we can also utilize the information from different platforms, different patients, to find the robust community. So this is important because spatial communities are new type of features. How do we know that those communities are not specific for one sample, but robust for one biological condition? Our approach is that we combine multiple technology, and we see in the x-axis here, and we use the cell type information per technology as the measurement to find the robust cluster or groups of communities. And one example of the melanoma community showed in red in here, and from that community we can ask again the question of which cell types are interacting based on the co-localization of the cell types. Very interestingly, now that the spatial cohorts that are assumingly always small, relatively compared to the population genetics study, which can be 500,000 people or over 5 million people. That is one of the biggest population genetic study published in Nature last year. So how can we combine that genetics information with the more mechanistic information from spatial data? So we apply this creative method from a college of us called GSMAP. And the principle of doing this is to look at gene expression data in the spatial tissue and correlate that genes with the SNP. Those are the DNA variants that are nearby the genes, in this case, plus and minus 50 kb from the genes. And from there, we can see, given the enrichment of the gene expression at one location in the spatial tissue, what are the heritability that can be stratified into that spatial location. And the effect of that is that we can map which cell types or which spatial categories that are most important in terms of genetics, heritability, association with the trait. And the trait in this case is the skin cancer. Applying that method, we can stratify the genetics association to spatial location. As expected, we can see that they are very enriched in the melanoma using the Visium data. Using the cosmic data, we can also show the higher enrichment in the tumor location and the immune location, but less so on the normal epidermis and stroma location. So this is a way where we can use the spatial data to inform back the genetics analysis, as well as use the genetics analysis to inform the spatial study to see which cell type and which location is important. And that integration now can bring open a new opportunity to detect the association between gene, SNP, and cell in the tissues. And that allows us to detect some of the missing candidates that come from lack of power in the population genetics data, but can be informed by the biological mechanism. So that is the part where we can see the integration of multiple modalities and multiple types of study that can inform the biology. So next, I present a few approaches that my team are very motivated to work on. That is to bring spatial to as many users as possible. So one of the approach is to use this method called on-slide tissue selection and transferring. What it allows us to do is we can select any shape or size of the tissue region. It can be based on the pathologist information to then transfer that standard pathology slide to the spatial slide. As many of us know that we are not allowed to get the blocks from the pathology unless some spatial arrangement can happen. In this case, we don't need the block, we just need the slides. And from there, using some special tools that can be printed 3Ds, we can get the shape of the tissue that we want, and we transfer to the spatial. Doing this, we can do like 50 samples per slide, and those samples are spatially informative, not relatively compared to tissue microarray. And the next way of bringing the spatial to more users is through computational tools. Many of the talks have and poster have mentioned the use of the H&E to predict the gene expression and cell types. We are very active in this area, and our focus are on estimating the uncertainty of the prediction. By doing that, we increase the robustness of how the model would work when we apply it to our sample distribution. And we also look at the interpretability of the model so that we can suggest that to clinicians at the time where it's applied. So I want to finish by introducing this consortium called Spatial Temporal Consortium, and many of us here have contributed to this consortium. The first meeting was last year in Singapore. So we got members from 37 countries, and we did some workshop to plan on how the consortium might be useful for the community. So the consortium is focusing on the temporal aspects as well as the multi-species aspect of the spatial study. And we try to tackle some of the scientific questions that is not possible to address by one group. For example, how can we look at the evolution of the multicellular in microenvironment that provide the complex functionality for that microenvironment? And we can do that through multiple species from lower to the higher end of the evolution tree. And I wanted to emphasize that there can be many consortiums, and the key thing there is how the consortium can work together. And in this consortium, we get some really useful and helpful advice from some of the senior members of the previous consortiums and existing consortium on how we can work together. And for example, sharing data, trying to answer the questions that are related but not overlapping. And so I presented to you the multi-omics approach to understand the biology of the skin cancer. And that approach can be applied to other type of disease or phenotype. And some approach to scale up the spatial data, bringing it to more users through experimentally the approach, experimental approach and analytical approach. And finally, the consortium also working on spatial, and that is open and really keen on collaborations. I want to end there by thanking my groups and collaborators, and thanks again for the chance to present our work.
Thanks, Quan. Really, really interesting talk. I think we have time for one or two quick questions. Joe?
Hey, Quan, right here.
Hi, Chef.
Great work, very exciting. Can you tell me a bit more about how you excise a certain part of your tissue and put it together? How stable do you have to be in terms of doing this manually? And are you doing this manually, or is there automated solution to multiplex?
So at the moment, we do it manually, and we are very careful about selecting the relevant part of the tissue through working with pathologists. The way that we take the tissue out from the slide is also very robust in the way that we have a sharp edge, and then we punch it and we take it out. And we have made comparing with or without transfer and compare the quality. And surprisingly, the quality is very similar compared to with and without transfer. By doing this, the time it takes to prepare the slide may be 2 hours extra, but the amount of information is huge, and the cost that we can save is a lot. And the convenience of getting the slide from pathology lab is a key when it comes to the logistic of acquiring samples and generating the data. Very happy to share. Yeah.
I think anything that can democratize it. If I understand correctly, you're putting lots of samples on small pieces on one slide that increase your cohort size. That's great. I was wondering, going one step beyond that, how do you make these spatial omics technologies amenable to international, cross-national collaborations?
Yeah, so we found out that shipping the slice or tissue section scroll is quite convenient. So as long as the ethics and material transfer agreement can be sorted out, this method of transferring the material across countries is very doable. Especially if we want to have one block and we want to share the material of that block to multiple collaborators at multiple countries, or we want to share a couple of slides from the same block. So It's very different to using two different blocks from same patient.
Thanks very much. I think this is a great opportunity for the community. If we can set up the MTAs and IRBs to be able to ship FFP samples or slides across countries, then we can create, grant proposals that will be very compelling to compare across ethnicities, across countries, and so on.
Exactly. And we are working with collaborators in Thailand and Malaysia and India as well on how to make this happen with the skin cancer as the first step.
Fantastic.
Thanks.
Okay, thanks again, Quan. So next we have Khanh Do. Is Khanh here?
Hello everyone, my name is Khanh Do. So today I'm going to present our recent work at Veneca University in the collaboration with Karolinska Institute about the Alzheimer disease. Especially we focus on the metabolomics and the signaling pathway in the Alzheimer disease. So this is just the overview for today's talks. So the Alzheimer disease is marked by the amyloid plaques. So, we usually use the tan phases. This is the stage to measure the A beta, the plaques spreading out through the brain, starting by the neocortex to the deeper regions, and then eventually reaching the cerebellum. And in parallel, the tau protein, they are hyperphosphorylated and become the tangles. And the brass stages is the stage to measure the processing of the tangles in the brain, starting from the entorhinal cortex and then spreading over the neocortex. And for the research, especially in the Alzheimer disease, we usually use the brass stages to measure the severities and the processing of the Alzheimer disease. So the lipidomics, the illustrator here is about the multiple omics. So the metabolomics is the part of the omics. So we study about the metabolites, and 75% of the metabolites are the lipids. So the lipidomics is the study about the lipids in the cellular lipids, and for the identification, quantification, to study about their functions in the health and in the disease. So the brain, the lipids, the brain is composed the 60% of the lipids by the dry weight. And the lipids has so many functions in the brain, not only the cellular structure, but also for the signaling, the intracellular signaling, and also the energy storage. And to study about the lipidomics, we use the LC-MS. So by these, the two phase. The first phase separating the chemical compounds by their physical properties and analyze them by the mass to charge ratios. So to study about the lipidomics in Alzheimer and also for the RNA sequencing in Alzheimer, we collected the samples from the Karolinska University Hospital. So the patient had to go through the deep diagnostic. And for example, we measure the CSF biomarkers and the genetics markers, the imaging, and also the MMSE score. And for the studies, in the CSF sample, we have like 174 participants by 3 groups, the AD, the MCI, like the early stage, and the SCI, the control. And for the human brain tissue, we collected at the brain bank at Karolinska Institute. So for the CSF samples, we show in here the lipid, like we found more than 500 lipid structure in the CSF. and it belongs to multiple class, like the lyso-PC, PC, PE, and SM. We show that the differences between the early stage, the control, and the AD. And for the human brain tissues, we found that multiple lipid mediators like the resolvins, the neuroprotectins, these mediators was discovered by Dr. Sahan from Harvard Medical School. And we show in the human brain tissue, the lipids mediators was also changed. So this table are the summary of the functions of these lipid mediators in different diseases. And to have the overview of the lipids in the brain, we perform the spatial lipidomics by using the MANDI mass spectrometry imaging. So we saw that in the APP mouse models, the mass A20 is the arachidonic acid containing phospholipid. It increased in the APP mouse model. And the mass A44, it's the DHA containing phospholipid. It decreased in the Alzheimer models. So we saw that the lipid, the alteration of the lipidome during the Alzheimer disease. So the next phase, we would like to study about the transcriptomics. So we perform the metadata analysis to build. First, we want to build the AD project by the multiple regions and as much patient as we can. And we address the alteration for each cell type, focusing on the lipid metabolisms pathway. And in the future, we want to collaboration, and we also did the support from the older neuroscientists to explain and further discover from the findings. So for the meta-analysis, we perform both the public, we curated both the public data and also combined with the in-house data, because Vietnam, we don't have any the single cell instrument yet. So all the data was performed in the US and at the Karolinska Institute. So for the public data, we do have the criteria to select the data. So first, we have the condition like the AD and the normal control, especially if we focus on the brush stage. And for the clinical information, we did the cognitive assessments, the mental status, and the post-mortem interval, and also we focus on the single-cell platforms. So we use the bioturing software to reprocessing the data and data analysis, and especially focus on the metabolism pathway. So for the overview of all the data that we have, including the in-house data, we chose the 10x Genomics platform because we use the 10x Genomics in the US. And the brain regions, we do have many brain regions. And for the overview, we collect the samples from the 6 brain banks over the world and the ROSMAP, the ROSMAP study. And for the information, we have analyzed 3.4 million cells from more than 600 participants, including 43 AD and 306 control from 7 brain regions. And for the clinical information, we have the quantity assessment, the mental status, the prostate, the amyloid score, the gender, age, and the race, mostly the white, and the post-mortem interval. And the UMAP here showing the meta-analysis of 3.4 million single cells. We focus only the main cell types in the brain. So for the each areas, we do have some, we focus on some important brain regions. So for the cell types, we focus on the main cell type like neurons, astrocyte, microglia, oligodendrocyte, pericytes, and the vascular cells, because we saw the changing, the alterations in the morphology, inflammation, and the cell death in this cell population. And for example, the microglia, it activated the morphology, revealing by also the immunostaining. So for the pre-processing, we did the quality control, normalization, batch correction, and the UMAP. And for the data analysis, we performed the cell clustering and the cell labeling by using the well-known markers for the each cell type. We acknowledge the complexity in the brain, and we aim to have the support from the Human Cell Atlas and also the CAP for the older annotation for older cell type and the subclustering in the cells. This UMAP is for the multiple brain areas. And then we we analyze the cell composition, the gene expression, the gene ontology for different brain areas. So for the cell composition, we saw that the cell composition varies across the brain regions. And in the AD, the increase the excitatory neurons in the prefrontal cortex and in the hippocampus. And the oligodendrocyte side is shown the reduced in multiple regions. So for the deeper analysis, we look at the excitatory neurons in the prefrontal cortex. We show in here the more than 200 upregulated genes and about 40 downregulated genes in the excitatory neurons, and they are involved in multiple pathways like the oxidative phosphorylation and the senescence and the ribosome functions. And we listing here the gene list that related to the metabolism pathway. It's such a very long table. So, and then we perform the GO. These genes are related to the lipid metabolic pathway and also for the signal transduction by lipids. And I categorize the genes by the GO. So in the conclusion, we know that the lipids are important for the brain structure and functions. And the lipid tools like the LC-MS and the MANDI, we can perform the spatial metabolomics in the brain and also for the older tissues. And the insight from the single cell, we review like each brain regions have the unique cell compositions. And in the Alzheimer, the metabolic pathway will change in the Alzheimer disease. And for the future direction, of course, we have to improve the brain cell type annotation with the support from the Human Cell Atlas. And for the metadata limitation, right now we just send out the email for the original orders to get the full metadata. And then we can compare the race, the genetic versus the sporadic Alzheimer comparison. And then we enhance the lipidomics resolution, especially for the MANDI, so we can perform the better resolution for the spatial metabolisms. And we would like to, in the future, we would like to integrate the MANDI for the metabolomics and the transcriptomics for further to build the comprehensive mapping for two omics. Okay, thank you. And yeah, and I would like to say thank you to my supervisor, Dr. Bazan in the US, and also Dr. Skupert from Karolinska and Dr. Sohan for the technical support. And especially thanks to Dr. Sun Pham from BioTuring for the support for the computational and analysis. So thank you.
Okay, thanks. That was great. We have time for a couple of questions. Yeah.
Okay, just quickly, I'm curious, what additional, did the lipidomics help you interpret the single cell data differently, or see something in the single cell data that you didn't notice before?
Right now, we just did the regular single cell RNA sequencing. So we found the lipidomics signaling will change at the transcriptomic levels. So that's why in the future we aim to build the spatial single cell hub in Vietnam, so we can further have a look at the integration between the lipidomics and the RNA sequencing at the spatial level.
Thank you.
Oh, okay. All right, thank you. So next we have Woong-Yang Park from South Korea.
Okay, thank you. For a regional network, actually, I want to introduce the Korean network for the single cell research. So mostly I want to introduce that kind of some research group, the collaboration, and also second one for infrastructure to support that research internally in Korea, but also in that I hope to communicate with other scientists in this room to utilize our data set together. Okay, first, actually, the history of our society in Korea is initiated from 2017. At that time, that we, sorry, actually, it's the 16th, the first meeting in the HCA, we joined in Israel for the first meeting. And the next year, we have the meeting in Okinawa in the HCA Asia. And at the time that we initiated that kind of some society we need in Korea. So we found that the research group under the Korean Society of Biochemistry and Molecular Biology as a research group. We have about more than 60 PIs join this kind of research group. So we have annual meeting for the single cell research internally for the just Korean research. And then in 2018, we luckily have the chance to organize the second HCA Asia meeting in Jeju. So yesterday we announced that, but actually in 2027 we will have another meeting in the Jeju HCA Asia. So I'm welcome to participate at HCA Asia meeting in Jeju. So in 2018 Jeju, we have initiated some collaboration between the Asian countries. So Jiang and Jay initiated PBMC study. Finally, we have some AIDA project. So at the time that we discuss about what we can do together. So I think it's a good chance to discuss these kind of things in HCA and also HCA Asia. So I hope to summarize that. Also, that we hope to have a second or third AIDA like the consortium together through this kind of discussion. And this is led by the members listed in the figures, and especially in the Jong-Il Kim and also Jong-Won. He led this kind of group. So I'd like to introduce two big project. Like one would be probably yesterday in the Jong-Il introduced that SCADE, single cell immune disease, Korea. So included multiple immune disease, and then it is important. I hope to link that the AIDA data, because we have now get the Korean immune features, and then we can combine with the disease data. And then the PBMC and then tissue immune, but we can combine this data together based on the AIDA Korean data together. The other will be not a research group. Actually, I hope to introduce another annual meeting held by Junil and Jongbin. Actually, young scientists, they actually organize nice meeting, international meeting. So I'm happy to invite you to join this meeting. Especially they organize annually hackathon. So for the students, also the scientists, they actively join this hackathon to learn the bioinformatics tools and the methods. So and then we also many scientists provide the data, and then so even my students, they learn a lot from this kind of process. And now switch to the database and infrastructure in Korea. As you know, we understand the immune diversity through the AIDA project. Also, we understand that we have some genetic diversity between Asian countries. So to have the genetic archive in the Korean population, that is important for understand the immune diversity too. But actually, we also keep working on this kind of study. So now we published the 5,000 whole genome data, but actually we will increase the number to provide the scientists in Korea, also in the scientists in the other world, to provide some unique features for the Koreans, the population. And then this data will be collected in the Korean database. COBIC and Korean Bioinformatics Center, they organize this kind of database. So you can, most of this data, maybe at least in the controlled access, you can get the data from out of that. So I deposit the colon cancer data into this database. So maybe you can get the data out of this COBIC KBDS. So Korean government supported this kind of the project to collect the data from the scientists, and then they can deposit and they utilize the data. And also we provide the Jong-won actually nicely make some annotation metrics, and so he provide this annotation method for the scientists, anyone who want to use this kind of things, and he collect a lot of the single cell data to, especially for the multiple cancer types. So, and then also he now prepared some spatial annotation method together. And also we collect the public spatial data. So maybe you can freely download the data. At this moment, we can open the 100 cases, but we now update about 300 cases. So including across the platform, so we can download the other type of data that is published, and then we say get permission to share this kind of data. So maybe you can freely get access to that. And also, at least you can look at the data through this database. So we introduce that kind of database to the bioinformatics journal. So last one, I want to introduce our job related to how to manage the quality of the spatial data. So when I discuss with the other groups in the other country, actually, it's not easy to. You can compare each other for the different data sets. But in AIDA, actually, we use the Caucasian reference sample for the same lot of the PBMCs, so we can distribute that to check the data difference between the countries, between institutions, different platforms. So I think this is my plan, my idea. But actually, because it's a xenium, so I think maybe we need some tissue. You can use that mix of cell lines to represent all the cell types. But actually, we need some architecture, to cell structure. So I want to use that some healthy normal tissue. But as you know, it's not easy to get the healthy normal tissue out of the donor. So, but actually, luckily we can get 3 donors for the liver cancer. But actually, we try to get the most distant region without any pathology. So we check that by pathology, and out of 3, maybe I can get 1, not completely normal, but actually apparently the pathology say that there's no the tumor cells, no immune cells infiltrated in that area. So we can dissect. We make it into 23 millimeter the tissue cube, and then make it as a block. And then we will distribute this kind of block for the other scientists for research, because this is healthy normal 1%. So I think maybe at least, as you know, the liver is relatively homogeneous. So I think maybe we can compare different block, but actually same donor. So I think maybe I have to compare each other. So I want to introduce the 2 cases. The 2 institutions, the different block, but the same donor. So I hope to check that how much the dimension of the diversity. Dimension of the variability between blocks and different institutions. Right. So we usually use the biopsy samples. So we bring about 10 to 12 the biopsy samples in one slide, gene slide. Then when we have some space, empty space, and then we can put in the 3 millimeter the liver tissue. And then we run 6 times, and then for 2 institute, 3 slides each. And then we get the data about 80,000 single cell for each experiment, and then we can compare each other. So one of the experiment, actually at the time that there's some problem in the genome machine, so we expected the data is not good, but actually as you see, second row be at the time there's the accident that would be happened, so we expected the data is not so good, but actually as you see the median value is low and compared to the other things. And overall when you compare with the 6 cases, I want to compare the top 100 genes. Roughly, we can say that about more than 80% of the genes, they overlap. That means 80% of the 83, 86 genes commonly found in the different experiments. It's not 100%, but actually maybe you can see kind of some variability between the samples and then expression levels. And then with up to 1,000, maybe it's more than 86% overlap each other. So institution A and institution B. So institution A and institution B be different blocks. So maybe that the distribution of the hepatocyte to be a little bit different because of different region. So maybe I did not look at detail, but actually it's some different region, maybe showing a little bit different patterns. So I think maybe another point we can analyze later. And also we look at the data together. We just compare with the reference sample, but actually relatively consistent, but actually we cannot see any difference, but some technical, experimentally the variability we can detect, but actually it's not so significant. But in this the figure, actually we visualize all the almost about 60 tissue data. And as you see, the gray line we represent the reference sample, liver samples. They show the consistent across the 6 samples. And the median level is almost same, but actually the second one, they show that, sorry, actually, I cannot see that. So anyway, so the one experiment is not so good, but actually the cancer sample usually show that nice pattern. But institution B show that very highly consistent across the cancer, including multiple cancer types, but actually still it's consistent. And then sometimes, yeah. But actually in the institution A, they actually show this wide variation in the tissue. So, but actually we can say the reference is same. So experiment process is, there's no problem at all. But actually some depends on the sample preparation, I think. So the difference between institution A and institution B, so actually also that we check that cell numbers. There's no, the quality is not, there's no the correlation with the cell numbers. Even the small biopsy sample, they show the nice data. Also in the big sample, sometimes it is not good, but there's no correlation for the sample size. And then institution A and institution B be different institution exactly, but actually institution B, they running core lab. So in the pathologist organize everything. So putting the tissue by pathologists and then technicians in the one single institute. Also, they control that SOP for the paraffin embedding, also formalin fixation conditions. So it's a kind of some nice code running for 10 years. So they use the sample, so they show that nice, the correlation, the consistent pattern for even in the different cancer types. But institution A, they receive sample from the outside, in the multiple institution, and then there's no control for the SOP for the pathology process. So as you see, as you expect that different institutions, different method, different protocol, different formalin fixation conditions, so maybe you can expect that the variability coming out from that. So just my speculation, but actually we can say that if you not control that, the paraffin, sample preparation for until you get make a paraffin block, the variability will increase. Okay, I want to stop here to introduce that to part, like research group inside in Korea. And also I did not mention today about the Asian Cancer Atlas. Actually, we tried to organize the HCA Asia members to share the cancer data together. So in the Thai and Japan and Korea and also in Australia, joining together, and also happy to invite the other members together. So anyway, so we are comparing the cancer immune for across the countries based on AIDA project. Maybe you can understand that cancer immune diversity in between Asian countries, because as you know, the cancer pathogenesis different each other because of same environmental or other immune system is different. So I think maybe you can find other interesting features in the cancer immune atlas. And also into the database infrastructure, I'm happy to, maybe if you have any experience in the problem in the download for the data from the Korean Bioinformatics Center, probably let me know, and then I can help you to download the data from the Korean populations. And also I'm happy to, it's not many, but actually I can happy to share that the liver reference block with you. Thank you.
Yeah, that was great. Thank you so much. Do we have any questions?
It was amazing to see that the technical, that you're integrating spatial data across sites. That's really a key, and so taking AIDA to spatial and multi-site. And I was struck by the fact that you're getting a lot of technical variation. Would you say this is more than the technical variation we had in the PBMC single cell data in AIDA across sites?
You mean the technical variation between from out of the AIDA data?
From sample to sample, you showed the QC specs being, for some samples were quite variable, right? Is that more than, say, in single cell data?
Right. Yeah, I mean, you mean the spatial data, right?
Yeah.
Yeah, I think maybe it's spatial data mostly in the cancer tissue. So it's not easy to say that some variation between patient or between countries, between institutions, because so the variation dimension is wider than normal heteronormal PBMC. So I think maybe at this moment I cannot say, but actually the institutional variation is relatively small. I think maybe it totally depends on the sample preparation process, I think, yeah.
Right, so you think mainly it's just coming from the surgery?
Yeah, yes.
Okay.
Okay, yeah, I think we're at time, so thanks again. And our final speaker for the session is Jiang, so welcome.
Yeah, good afternoon. Thanks for the invitation. It's my great pleasure to be here to present our work. So I'm Jiang Lan from China National Center for Bioinformation. So our center is in Beijing. Previously, we have another name is Beijing Institute of Genomics. It's affiliated with the Chinese Academy of Science. So we all know that this international consortium is the reason we are here. But in my opinion, even this consensus reference map is done. It's definitely not the end, but it's a beginning. In the future, what we will do is personalize cell atlas. The term may be not still in the progress to discuss, but I think the concept should be established. We really need to think about that. What's the new those single cell and spatial technology can bring us compared with the traditional genetic sequence. And I would like to highlight two aspects. The first one is we can do patient stratification based on subpopulations. And second one is we really can investigate the origins of disease and promoting early detection of and interventions. However, currently there's the high cost of single cell sequencing remains a critical barrier for scaling single cell sequencing to populations scales. We know Nico and Joseph, they both have done amazing work that construct those large-scale studies. But the size of this type of studies is still rare because there is a high cost. So our lab is focusing on develop some new technologies that can increase the throughput and reduce the cost. And the strategy we use is called droplet microfluidics combined with combinatorial indexing. So previously we have already published a work named FIPRC, and it can achieve ultra high throughput of 100K cell or nuclei per channel using existing commercial platform. And what's really new is why it's a novel approach is you can see illustrated in this slide. For the sign line is really show you that the correlation rate means two cells have the same cell barcode is an error. This correlation rate is really proportional to the throughput of the droplet microfluidics. That's why there's a throughput limit for the platform. However, after we introduce combinatorial index to droplet microfluidics, the red line shows that the correlation rate is dramatically reduced. That's why we can increase the throughput, and theoretically it can be even higher throughput. But typically we just use to generate the data for 100K cell per channel. And additionally, we have made an update for these strategies we call the AIDA-seq. And the new thing is we upgraded to, we switched the order. We do not do pre-indexing, but change to post-indexing. Now this strategy can support multimodality core assay from the same single cell and achieve ultra high throughput, nearly 100,000 cells per channels. We have it's already published. We have the data to demonstrate that it really can support core assay of gene expression and VDG sequencing and co-expression, co-detection of gene expression and content stability, and also co-assay of gene expression and CRISPR interpretation. So the take-home message is that they really can reduce the cost, especially for large-scale project. I want to highlight with those powerful technology in our lab, in our hand. So I want to highlight 3 projects that currently is ongoing by collaboration in China, in Beijing. So first one is we collaborate with the physician of renal disease. We construct a single-cell multi-omic atlas from more than 200 donors, have 16 diagnosis categories. So the diagnosis is made by the doctor in the top-tier hospital in Beijing. And we only take 10 channels of multi-omic by AIDA-seq strategies to connect more than 1 million cell data. And each cell have the gene expression and the content stability simultaneously. And we will generate a panel of cell type specific genes and the pathology associated genes and also key transfactors to form a spatial panel to make really push the diagnosis in renal disease to the next level. And second one is we construct we study the aging immunities by this time by integrating the gene expression and VDJ sequencing. And again, we just take us 10 channels of HCA to generate 1.18 million cells, and those data is generated from cohorts of aging cohorts, the aging from 20 years old to 84 years old, and from more than 200 donors. And what we do is really want to build a multimodality single cell data set to make a model to predict biological ages. And this model can use to evaluate the different anti-aging interventions to make individualized suggestion for anti-aging interventions. And the third project is we apply HCA again is for gene expression and content stability co-assay to study the inflammation-driven oncogenic trajectory in cholangine carcinomas. This project, the design is including some healthy control and inflammation status, and also some pre-cancer samples, and also including the major subtype of traditional pathology subtype of CCA. And we want to understand what's the critical transition point between the pre-cancer to carcinoma. And we also want to classify based on the cancer cell or microenvironment to make the suggestion for the treatment for different subtype. Okay, so the HCA, the published one, we do 2 rounds of barcoding, but it's rely on genetic variant to do sample multiplexing, so it have some limitation. So furthermore, we have made another update. It's called HCA2. In total, we now will do 3rd round of barcoding, and including both pre-indexing and post-indexing. And the advantage is now we can further increase the throughput to stably have 300,000 cells per channel, and also the pre-index step will can achieve sample multiplexing, which is independent with genetic variant. And we have we have applied this HCA2 to 3 settings, including 2 non-human organisms, including mouse and development and non-human private aging. And the third case is interesting. We apply this HCA2 collaborate with a drug company. They have the first approved mesenchymal stromal cell drugs in China, and they want to evaluate the cell status for every 4 hours during the manufacture of those cellular drugs. And we apply HCA2 to kind of do the cell identity assignment to those cellular drug to prove the drug is the same cell for different batches and derived from different donors, which is the same as they used in the clinical trial. Okay, so the HCA is not we have approved it can support the multimodality of 3 assays in the upper panel. We further expanded the same strategy to achieve ultra high throughput of gene expression of other epigenetic modifications, including histone modification, DNA modification, and M6E modifications. And we already have the data to demonstrate that these strategies really work. Okay, so we want to emphasize that EUDA-seq is not just a proof concept. We really already applied to real-world large-scale studies, and so far EUDA-seq have generated more than 5.6 million cells from nearly 1,000 donors across more than 10 tissue types. And we also established a mechanism during the collaboration that so my center is actually the place in China hosts all the genetic data in China, and by our technical innovation, we have a lot of collaboration ongoing, and we try to also make some innovation of mechanisms to make those data generated by us also can be shared with other groups, so we will contribute to the data generation, and the collaborator, most likely physicians, they will contribute to the cohort establishing and cover the cost. And we have agreement to share the data, or of course, we can reshare the data, but with control of access. Okay, the summary is EUDA-seq is integrated the existing droplet microfluidics, so to achieve ultra high throughput multi-omics, and we furthermore, as our workflow is compatible with clinical sample from frozen needle biopsy and frozen PBMCs, and we really want to facilitate cohort studies for typically 100 or 200, if the sample size is 100 or 200, even a single lab actually can make it affordable to average lab. And I didn't have time to go to the detail. Actually, we during the data analysis, we found a lot of interesting link between certain subpopulation to the phenotype we care about. And I want to, in other aspect is with this large amount of data, our group also working on build a new algorithm or models. So this model called cell memory, we leverage a bottleneck the transformer architecture that have strong generalizability and high computational efficiency, and we use this model to do population scale single-cell data integration and also granular level annotations. And the interesting part is we also use this model to inferring auto-distribution cell, for example, compare disease versus healthy reference, or compare spatial data to single-cell data reference. And our model have a hierarchical interpretivity, including gene level and gene program level. And my student Qifei Wang have a poster yesterday to introduce his work. Okay, I don't have enough time to go to the detail, but another point I want to emphasize is that we really observe that for different patients, we found that their cancer cell is derived from different lineages for leukemia and multiple myeloma. We have those new technique and new algorithms. So we combine these two kind of work together. Now we are working on build a multi-omics foundation models for a specific organ. So we have in-house data, multi-omic data, and we also collect the public available data and use this bottleneck transformer architecture. We build this to our knowledge, the first multi-omics foundation model for this organ. Yeah. Okay, I want to thank the key contributor in my lab, Li Yun, Huang Zhen, Wang Qifei, and also the collaborator and the funding agency. Thank you.
Okay, thanks. Really amazing methods. And actually, we've been running AIDA in my lab as well, so yeah, we're big fans. So, any questions for Jiang? Maybe I'll start. So the aging PBMC study that you mentioned, I'm curious if are you looking at clonal hematopoiesis in that as well?
Hematopoiesis?
Yeah, clonal hematopoiesis.
You mean from the bone marrow or from?
Yeah, because you mentioned this aging PBMC study.
Oh, yes.
Yeah.
It's from PBMC.
Right.
Yeah.
Is that something that you guys are looking at? Maybe there are other methods of identifying somatic mutations through looking at, say, mitochondrial variants and these sort of things. Is that something that you can do with the type of data that you're generating?
So our analysis focus is more focused on the transcriptome and the clonal expansion, AG related.
Okay. Yeah. Thanks.
Yeah. Thank you.
Yeah. Yeah. Go ahead, Jeff.
Yeah. So great work. You mentioned about data sharing, and I understand that it's for the Human Cell Atlas community here. Can you share a bit more about? Is it possible to have managed access to some of the data that are generated in China, and what are some of the ways that we can start the conversation about data sharing globally?
Yeah, thanks, Jay, thanks for the question. Yeah, that's definitely our goal, at least for myself, our goal to achieve that, to do, data sharing across the different country. We want to do that because that will make the data more valuable. However, it's currently it's still ongoing, but we have already taken a step, move forward, and we, at least for the data generated by our collaboration, we will require the data is uploaded to the GSA database, and people can see it and people can apply. But the data contributor means the physician that they have the right to refuse to share the data, but it's case by case. It depends on which team is applied, which team, what's the purpose for user data. Yeah, that's the current status.
Is there a barrier to sharing expression matrix? I understand.
Oh, no,
data.
no.
Okay.
Yeah.
Great. Thanks.
Yeah. Thanks.
Okay. If there are no more questions, let's thank all the speakers once again. And we'll meet back here at 3:40 for the second part.